Thank you. Thank you so much for a great talk. Moving on to introduce our next speaker, Dr. Gyeyoon is a postdoctoral research fellow in the Environmental and Occupational Medicine Epidemiology program at the Harvard T.H. Chan School of Public Health. She earned her doctoral degree from the Harvard Chan School in May 2020 with a dissertation entitled Periconceptional Exposure in Child Health in the Third Generation. Dr. Gyeyoon's main research interest for her doctoral dissertation was to investigate how grandmothers' exposure to external or internal factors could have a multi-generational impact on their grandchildren's risk of neurodevelopmental disorders, such as attention deficit or hyperactivity disorder, and adverse birth outcomes. Previously, Gyeyoon obtained her master's degree in social and behavioral sciences from Yale School of Public Health, worked as a researcher at the Korea National Institute of Health, and served as an associate specialist in the Department of Psychiatry at the University of California, San Francisco. So now, Dr. Gyeyoon, I will turn it over to you.
Yes, so can you hear me well?
Yes.
Perfect. Yeah, so thank you so much for your nice introduction, Rose. And then, hello everyone, my name is Gyeyoon Im, and as Rose just mentioned, I'm currently postdoctoral research fellow at Harvard T.H. Chan School of Public Health. And today I will introduce the main findings from one of my dissertation papers on the association between smoking during pregnancy and risk of attention deficit hyperactivity disorder in the third generation, which means grandchildren. So first, ADHD. As you might already know, ADHD is one of the most common neurodevelopmental disorders, which is characterized by inattention, hyperactivity, cognitive impairment, and impulsivity. And the prevalence of ADHD is approximately 7% worldwide. And as you can see from this graph on the right, the prevalence of ADHD is on the rise. And when it comes to smoking during pregnancy, even though the prevalence of smoking has declined over the past decades, but still 7% of women in the US are still smoking during pregnancy, according to CDC in 2016. And as you can see from this picture, the smoking during pregnancy not only affects the pregnant mother and also the fetus, but also it may induce multi-generational DNA damage or germline mutation. So I guess maybe one of the questions that might naturally come to your mind might be, why are we linking ADHD and smoking during pregnancy? So given the early onset of ADHD, scientists have focused on the prenatal risk factors, including maternal age at childbirth, prematurity, and low birth weight, and also exposure to environmental toxins, which includes smoking during pregnancy. And actually, in a lot of in quite number of epidemiological studies in the past, they actually found and they actually concluded in the most recent meta-analysis that there might be positive association between maternal smoking during pregnancy and the risk of ADHD among their offspring. However, unfortunately, the etiology underlying the ADHD is still unclear. So these days, pretty recently, scientists actually began to question, as you may have already seen in the previous awesome presentations, that they began to ask a question, there might be missing link. So there might be some like risk factors even beyond the maternal level. So which means like a potentially grandparental level. So there might be, so grandmaternal or grandpaternal smoking during pregnancy might affect the grandchild risk of ADHD, either via direct pathway or like a mediated pathway. So given these backgrounds, in the current study, we first investigated the association between grandmothers smoking during pregnancy and risk of ADHD among the grandchildren. And given the nature of this multi-generational association study, it was inevitable to examine how much of the observed association could be explained by the maternal smoking during pregnancy, which is one of the key potential mediators. So in order to conduct this study, we used the data from the Nurses Health Study 2, which consists of 116,000 female registered nurses in the US. And we also used the data from the Nurses Mothers Cohort Study, which was one of the sub-studies of the Nurses Health Study 2. Before I go further, I will briefly introduce the terminologies that I will be using throughout my presentations from now on. So as I already mentioned, if a woman is pregnant, basically all the three generations, the pregnant mother and the fetus, and also the reproductive cells of the fetus or the seed of the grandchildren, they are all simultaneously exposed to the environmental toxicant. So in this current study, I will now call the nurses mothers, which is in this picture indicates the pregnant mother as G0, generation 0. And the fetus will become the nurses, and I will call them G1, generation 1. And reproductive cells of the fetus, as they are now become the nurses' children in the current study, and then now I will call them like as G2, generation 2. And it's pretty much similar to F0, F1, F2 in the animal studies, but we just use the different alphabet in the human study. So to move on to the exposure and outcomes of interest, the exposure of interest was grand-maternal smoking during pregnancy, which was reported by not only the nurses but also the grandmothers, the nurses and mothers themselves. And outcomes of interest was grandchildren's ADHD diagnosis reported by the their nurse mothers. And in data analysis, we mainly use the cluster-weighted generalized estimating equations in order to simultaneously address the potential correlations among the study participants, as well as the potential issue of informative clustering with a logistic link. And we also further adjusted for the potential confounding factors. So before I get into the details of the main findings, I will first present the key characteristics of the study population. First, approximately 25% of the grandmothers smoked during pregnancy. And we also found that if the grandmother smoked during pregnancy, they were more likely to have had higher socioeconomic status in terms of college education and as well as the professional occupation status. And surprisingly, as expected, we also observed the higher prevalence of a lifetime history of depression among the grandmothers who smoked during pregnancy. And we did not really find any substantial or significant differences in terms of the distribution according to the grandmother's smoking during pregnancy in other variables such as race, ethnicity, or birth year. So we didn't really present here, but we made sure to adjust for these potential confounding factors in our main analysis and also secondary analysis. So on this slide, it shows one of the main tables of my study findings. So, and basically we investigated the association between grandmothers and or mothers smoking during pregnancy and the risk of ADHD among the grandchildren or their offspring, respectively. So to briefly explain about this table, on the first column, you see the specific exposure categories of the grandmothers or mothers smoking during pregnancy. And on the second column, you basically can see the number of grandchildren who were exposed to grandmothers or the children who were exposed to their maternal smoking during pregnancy. And on the third column, you may also see the number of ADHD cases according to each grandmother's or mother's exposure status, and also the proportion of each. And on the bottom, we also listed the potential confounding factors. They were selected a priori, and then basically the results from the unadjusted and also adjusted models were pretty much similar. So in this table, we are only showing the results from the adjusted models. So first in model 1 and in models 1 and 2, we basically just treated grandmother or mothers smoking during pregnancy separately. So first in model 1, when we only considered grandmothers smoking during pregnancy in relation to the risk of ADHD in the third generation, the grandchildren, we found a positive association, which was significant. And similarly, also in model 2, when we only considered maternal smoking during pregnancy, we found a positive association. But interestingly, the effect estimate was slightly lower than the grandmothers smoking during pregnancy. And next, in model 3, when we simultaneously adjusted for the grandmothers and mothers smoking during pregnancy in order to examine if how much of the association that we observed in the grandmothers smoking during pregnancy in association with ADHD risk in the grandchildren can be explained by mothers smoking during pregnancy. And we actually found that basically compared to the models 1 and 2, we actually found that the associations observed in the previous models remained pretty robust in both grandmothers and mothers smoking during pregnancy, and which indicates that there might be individual and independent biological pathways linking the grandmothers or mothers smoking during pregnancy to the risk of ADHD among the grandchildren or children respectively. And as one of our secondary additional analysis, we also examined the association between grandmothers' number of cigarettes smoked during pregnancy and the ADHD risk in the grandchildren using the data from the Nurses Mothers Cohort Study. And this graph basically shows the 3 exposure categories of the grandmothers smoking during pregnancy as a non-smoking group, and then 1 to 14 cigarettes smoked per day group, and then 15 or more cigarettes smoked per day group. And each column actually contains 3 bars, which represents each model, unadjusted, and model 1 and model 2. And then only difference between model 1 and model 2 is whether we further adjusted for the grandmother's lifetime history of depression. And as you can, and on the bottom, you can also see the small tables, which was pretty similar to the second and third column of the previous table. So basically, for each exposure category, you can see how many number of grandchildren were exposed to, as a germline, exposed to grand-maternal smoking during pregnancy, and also similarly, how many of the ADHD cases were we observed for each grandmother's smoking during pregnancy exposure category. So basically, in a nutshell, first, when we adjusted for the potential confounding factors, we found that the effect estimate is slightly attenuated after adjustment, but still even the adjusted models remained pretty robust and then significant, statistically significant. And as we move from the non-smoking to 1 to 14 cigarettes per day to 15 or more cigarettes per day groups, we were able to observe kind of nice dose-response relationships, which suggests that if the grandmothers smoked higher number of cigarettes per day during pregnancy, they are slightly more likely to have had grandchildren who are diagnosed with ADHD. So to summarize, we found that grandmothers smoking during pregnancy was associated with an increased risk of grandchildren's ADHD, which was independent of maternal smoking during pregnancy. And we also detected a nice dose-response relationship with regards to the number of cigarettes smoked per day. So in conclusion, given the results from the current study, the exposure to environmental chemicals during pregnancy may affect the development of the 3rd generation, which indicates the multi-generational associations. And given the possible germline effects of the environmental toxicant, it should be now evaluated from a regulatory perspective, hopefully in the near future. So before I conclude my presentation, I'd like to thank all for your attention, and then I'd like to send many thanks to my advisor, Dr. Mark Weiskopf, and other co-authors of the current paper who guided me through this research project. And also, importantly, I would also like to send many thanks to Beyond the Genes and Jill, who gave me this wonderful opportunity to present my study. And I will welcome any questions now or at the end of the presentation, at the end of the session. Thank you.